Okay, so thank you very much for giving me this opportunity to present my recent work today. So I'm Seiichi Ishida from National Institute of Health Sciences and also from the Sojo University in Japan. So today I'd like to talk about the current situation and the future direction of Japan under the title NAM's Regulatory Needs in Japan. So first, a brief self-introduction. So I previously belonged to the Pharmacology Division at National Institute of Health Sciences here. And I'm currently continuing my research at Sojo University. I'm also continuing my activities at NIHS as a visiting researcher. In addition, I serve as the chair of the Japan NPS initiative. We launched this initiative 2 years ago as a voluntary forum to discuss NPS development in Japan. Our goal is to build a community where researchers, industry, and regulatory stakeholders can discuss NPS across sectors. So I would also like to introduce the IMPSS Asian Pacific Regional Chapter. This chapter functions as a network for researchers and stakeholders of NPS in the Asian Pacific region, providing a framework for internationally sharing regional-specific challenges and initiatives. Currently, I serve as the co-chair of this chapter along Dr. Gu from China. Let me briefly introduce my own research background. My research interest is to reproduce microenvironment of hepatic sinusoids in vitro and to construct a test system that can use for evaluation, efficacy, and safety. Specifically, I'm conducting evaluation of drug-induced liver injury using MPS. So establishing an evaluation system for biliary excretion and developing model for MASH, metabolic dysfunction associated to steatohepatitis. To reproduce this complex microenvironment, the concept of 3D culture and MPS are essential. And this is today's agenda. Today, I'll talk mainly about 3 topics. First, the background of NPS development in Japan and the regulatory a trend of the regulatory authorities. Second, the importance of the inline monitoring to ensure the reliability of the evaluation system. And third, if I have time, the utilization of the artificial scaffold biomaterials to keep the evaluation system simple and improve reproducibility. As the first topic, I will explain the background of NPS development in Japan, driven by a strong need for alternatives to animal testing in the industrial sector. Japan is promoting the research and development of the development of new test methods through collaboration among industry, academia, and the government. So this slide illustrates the pathway toward industrial implementation and the further regulatory acceptance of NPS using the flow of the domestic projects in Japan. As an example, generally, for a new test method to be accepted by regulation, it is necessary to go through the stages of definition of the test method, confirmation of the reproducibility and usability necessary for the industrial implementation, and construction and the standardization of evaluation framework on the regulatory side. In Japan, the research and the development of test methods were promoted by the AMED NPS-1 project, which started in 2017 with the support of the Ministry of Economy, Trade and Industry. Subsequently, in MNPS2 from 2022, we are working on improving working on improving usability and reproducibility toward industrial implementation. In parallel with these, the MPSRS regulatory science projects led by the National Institute of Health Sciences under the support of the Ministry of Health, Labor and Welfare is bringing the gap to regulatory use through the standardization. In other words, the generation the great future of Japan is that the design and proceeds with the process continuously from research and development to industrial implementation and ultimately regulatory acceptance through regulatory science without fragmentation. As outcome of such MPS projects, multiple MPS platforms are already on the market or are in the stage just before the practical application. Today, I will introduce three products as example. The first is the Fluid 3D Cross. This is an X-shaped platform with apical basal surface that allows perfusion above and below a membrane, and it is highly suitable for examining the application of shear stress and the co-culture of epithelial cells and endothelial cells. The second is the BioStrat plate. This is an organ block type platform capable of connecting up to 4 organs by combining with the general purpose culture equipment such as the culture insert. A built-in stirrer type pump realizes simple and stable perfusion culture. Also, because it is an open system MPS, it boosts the high usability equivalent to the standard culture dishes. The third is the bone site. This chip features a structure in which porous membrane is sandwiched between upper and lower microchannel. By forming an epithelial cell layer on the membrane, it can be used for the pharmacokinetic studies and toxicity evaluation. All of these were developed by Japanese advanced microfabrication technology and pursuing easy of use of users. So regulatory authorities in Japan, particularly the Pharmaceuticals and Medical Devices Agency, PMDA, are also actively moving toward the implementation of new approach methodologies. The PMDA recently published a document titled Early Consideration, which begins to outline the guideline principles for the use of NAMs in new drug application. Furthermore, a cross-sectional working group for NAMs has been established within the agency. They are continuously discussing various issues and exploring the potential application of NAMs in collaboration with domestic organizations such as the National Institute of Health Sciences, CIVM, industry, and academia, as well as the foreign regulatory authorities like the FDA and EMA. As one of the case studies for regulatory acceptance, we are advancing an OECD test guideline proposal for alternative methods in the human health impact assessment of chemical substances. What we are proposing is a new concept of in vitro toxicokinetics. As the composition will be shown on the next slide, the evaluate and predicts the first-pass effect from the absorption and the metabolism of an orally administered chemical substances in the small intestine to its metabolism in the liver and entry into the systematic bloodstream using an in vitro two-organ MPS system. Then, regarding the prediction of subsequent systematic exposure, the approach is to conduct risk assessment by integrating it with a PBK model in silico model, for which revision work is currently underway at the OECD, the test guidance 331. As the development of the in vitro toxicity evaluation system for each organ progresses in the future, toxicokinetics will become the key for weighting and integrating these hazard evaluation based on the exposure rather than treating them separately. So I will explain the construction element of our proposal small intestine liver toxicokinetic MPS model. As small intestine epithelial cells, human iPS cell derived FI-seq manufactured by Fujifilm is available. Also, cryopreserved human hepatocytes are widely used. By combining the cells with Japanese MPS devices introduced in earlier or a commercial MPS from overseas, a test system for evaluating the first-pass effects can be constructed. This is the roadmap to the OECD test guidelines. The toxicokinetic evaluation using the small intestine liver NPS proposed by Japan was approved at the OECD WNT meeting last year. Currently, experts are gathered and proceeding with the creation of a detailed review paper, DRP. In parallel with this, domestically, under the support of the AMED projects, a multi-facility pre-validation test led by NIHS with participation of development companies, CROs, and JACVM is in progress. We believe that this proposal will serve as the touchstone for NPS proposal coming from other countries in the future. Japan is accumulating experience toward the development and the practical application of MPS through case studies such as the small intestine liver to organ models introduced so far. The documentation of this process and the development of new test methods is currently under discussed at ISO. This slide shows that the chart of the discussion on its development process. First, based on the context of use and the need defined by user, the overall performance standard of MPS is determined. Because MPS consists of the cells and the device, the criteria for cell and the standard for device are prescribed respectively to meet performance standard. Then, consideration point to clear the criteria are specifically derived, and all elements must be designed to meet the COU. We have derived these consideration points through case studies and are currently working on compiling them into the paper to be published in the English Journal of the Japanese Society for Alternative to Animal Testing. So based on these experiences, today I would like to make two proposals. The first is the introduction of the inline monitoring, and the second is the simplification of the evaluation system utilizing artificial scaffold. First, I will talk about the inline monitoring. So as a simple example, let's consider the supply of nutrients and the accumulation of the waste products in the culture. In general, in vitro tests, cells are seeded in a dish and placed in an incubator. Researchers often basically leave it to the incubator except during the observation or medium change. However, with this, the state of the cell can only be grasped intermittently, and it is impossible to know what is continuously happening with the medium. Actually, we measured the transition of glucose and lactate concentration in a standard static culture of HepD2 cells. This graph shows the result of the 120 hours of culture. As you can see, glucose is rapidly consumed over time, and at the same time, lactate accumulates in the medium. The change is particularly remarkable between 48 hours and 72 hours. If an assay were to be conducted using cells after the 72-hour mark, could those cells really be said to be in the healthy state? This data clearly shows that even in the standard static culture, the environment changes significantly and the cells are exposed to various stress over time. So, what about the case of the culture using MPS? In MPS, the medium is perfused using the pump supplied from the inlet and discharged from the outlet. The pump enables a continuous medium supply and waste removal, but the environment in which the cells are placed will be greatly affected by the continuous condition of the medium flow. This slide compares the concentration of glucose and lactate at the inlet and outlet with the MPS under two flow rate conditions, 0.5 microliter per minute and 2.0 microliter per minute. Under both flow rates, glucose is decreased and the lactate is increased at the outlet compared to the inlet. However, under the condition where the flow rate is increased to 2.0 microliter per minute, the range of change is smaller. That is, it shows that the higher flow rate allows the more efficient nutrient supply and waste removal, thereby suppressing environment fluctuation. Flow rate is a critical parameter directly linked to the metabolic state of the cells. The effect of the flow rate also clearly appears in the cell proliferation within the chip. This shows the number of the viable cells within the chip. Overall, the number of the viable cells is higher at 2.0 microliter per minute. Furthermore, under the 0.5 microliter minute condition, spatial heterogeneity occurs where the number of the cells near the inlet is greater than the near the outlet. If the flow rate is not appropriate, the state of the cell will differ even within the same chip. So changing our perspective, let's consider non-clinical animal testing. In animal testing, would there ever be a case where the animal is dosed in an environment without being giving enough food? Under the framework of the good laboratory practice, feeding, housing, environment, and observation items are strictly managed. It is precisely because there is the thorough observation and recording that the reliability of the test result is guaranteed. Then, how should current MPS be designed? Is it sufficient to simply trust the setting of the incubator and the pump? How should a record during the test period be managed? From a comparison with animal experiment, we reached the conclusion that the continuous monitoring and the recording of the deviations of cell culture are indispensable in MPS as well. MPS must be designed not just as a culture vessel, but as a test system capable of explaining what is happening inside. This slide summarizes the key items online monitoring necessary to maintain a healthy MPS. This includes continuous monitoring of the culture environment, such as the oxygen and pH, changing over the time in the metabolic state, such as the glucose consumption and the lactate accumulation. So these serve as the basic indicator showing the healthiness of the culture state. So I'll skip this one. So yeah. So this is the summary of this section. I propose that it is essential to introduce incorporation such as inline cell vital monitoring system into MPS. We need a system that constantly monitor the environment and keep logs during the testing period. As NAMs are integrated into the non-clinical safety testing, I believe that submitting logs of the observation and recording during the actual use, similar to the animal experiment, will become the most important point in the future. So it's almost the time is over, so I'll skip the third section and go to the conclusion slide. So finally, here is the today take-home message. In order to meet the strict context of use required by the FDA's stand program, the evaluation system must not be a black box. By visualizing and recording the cell environment through inline monitoring and by designing the system simply using the biomaterials, we will, for the first time, to be able to scientifically prove to regulatory authorities that it is fit for purpose. Japan intends to advance the development of next generation MPS that implement these fundamental technologies and contribute to industrial standardization. Thank you for your attention.